talk titled AI in GI, using AI in inflammatory bowel disease research.
Thank you. Thank you, and sorry for that very long introduction, but I'll get right to it. So thank you so much. It's really a great honor to be here, and I'm very excited to have this discussion with you. These are my disclosures, including grant funding and consultations, but none of this is related to the work I'm doing. The last point I think is very important. In contrast to, I think, many of the other speakers, I'm a clinician and a basic scientist. I'm not an AI expert. I'm not a computer scientist. So I hope to kind of give you my own perspective on this all. So who am I? This is where I started my academic career. This was my first paper, and the details aren't very important, but it came from a very clinical point of view. And this was done when I was a resident in Israel. That's where I'm originally from. And then I was very fortunate to go to Toronto for a clinical fellowship. And that's where I discovered the basic sciences, and I ended up doing a PhD in cellular microbiology, looking at the interaction between microbes and host cells in a very focused area that was not directly related to my clinical work. But when I looked for a job after doing a postdoc and then ended up here at the University of Alberta 13 years ago, it was at that time the microbiome was getting a lot of attention, and that's the area that I grew into on the research side, which did become very closely related to what I've been doing on the clinical side. So that really raised a need to understand how to manage big data, and that's really where I became introduced to AI, and that's what I'll talk about today. So I'm going to start with a very brief explanation on what inflammatory bowel diseases are, and the S there in diseases is very important, and I'll make that point later on. I'll talk a bit about some of the problems that we have as clinicians and in researchers in this specific area. I'll discuss some of the opportunities where I think that AI might be able to fit in. I'll then discuss two projects which are at very, very early stages, and in contrast to the two previous talks where we did see data from their work, we're not really at that point yet. So we're just starting to build this, and I'm hoping to stimulate some discussion around that, and I'll end off with some of the challenges and future perspectives. So inflammatory bowel diseases are a group of conditions that include the two main prototypes, Crohn's disease and ulcerative colitis. They involve chronic inflammation in the bowel, and the causes for IBD are really not clear, but it is a multifactorial complex condition, which I'll talk about in a bit more detail. The symptoms that patients might experience are shown over here. Generally, we divide them into intestinal symptoms, such as pain, diarrhea, and these can be quite profound and have a huge impact on the quality of life. I treat children, and children, we have some additional symptoms that we deal with. A lot of these relate to their growth, development, puberty, and I'm going to show just a couple of examples to give you the flavor of the patients that I treat. So this first patient is a patient who ended up having small bowel involvement. So the diarrhea was not a major part for her. It was more the growth and weight that were the presenting symptom, and she was found to have Crohn's disease, and this is what it looks like on colonoscopy. This is her small bowel. And the second case is a younger child with a much more brief history where the large bowel is involved, and he was eventually diagnosed with ulcerative colitis. And this is just to give you a very quick idea of what it is that I do as a clinician. So when we deal with a condition like inflammatory bowel diseases, I think it's really important to recognize the complexity, the multifactorial nature of this condition, and the heterogeneous features that really become a big challenge for us. And when we look at the potential causes, so about 20 years ago, some of the first papers came out identifying genes that are involved in IBD. This is a seminal paper from a decade ago that identified at that time 163 genes that were correlated with the risk of developing IBD. The number is now above 200, but this paper was really instrumental in helping us direct our research into the area that I ended up in, and that's the host microbe interaction. And this is taken from the title of that genetic paper, identifying that that's where the issues really are. So genetics have brought us to a certain point, but the next slide really shows us how genetics are really a very small part of the story. So this is showing the increasing prevalence of IBD, and in Canada, by the year 2030, we anticipate that 1% of the population will be living with either Crohn's disease or ulcerative colitis. On the left-hand side, you can see the total prevalence. On the right, the prevalence in children, and this is seen across the entire country. We have, in fact, one of the highest incidence and prevalence in the world with IBD, and interestingly, Alberta is really leading that together with Nova Scotia in those numbers. So there is something going on here that genetics can certainly not explain, because genes do not change that quickly. This is not just seen in Canada. This is a review paper that was published recently by a Canadian group showing across the globe in children where we're seeing the highest rate of increase. So most patients are diagnosed as adults, but 25% are diagnosed as children. And this is where we're seeing the highest increase. This all points in this conundrum of the pathophysiology of disease to the role of the environment, since the environment is what's certainly explaining this dramatic increase. One of the areas that I won't go into too much detail, which is probably the main recent focus of the lab, is how nutrition fits into this. But with this, I just hope that I gave you a bit of an idea of the complexity of the disease. So how does this translate into the world of treatment and what we're actually doing for our patients? I'm showing here 4 different aspects that relate to treatment of IBD that I think will maybe exemplify why this is such a challenging field. So on the top left, you can see the classic treatment pyramid that we've practiced over the years, where you start with a treatment. If it doesn't work, you move up to the next stage, with surgery being at the top of that pyramid. And that really makes sense. But over the last 15 years or so, with the availability of new treatments, we've recognized the importance of identifying patients who are at higher risk and taking what we call the top-down approach, where we actually start with the therapies that are usually kept for later on after you fail different treatments. And then we hope that we can step down from that. Now, at the top of the pyramid is surgery, and this is really an interesting change just over the last couple of years. I just came back last week from the main IBD meeting in Europe, and there actually are attempts to identify patients that will benefit from getting surgery right at the beginning without medication. And in a subgroup, that seems to be the right way to go. Now, the reason I'm telling you about this is that we have huge challenges in identifying which patient will benefit from each one of these. If we are providing a treatment, and we'll look at the bottom left over there, what's appeared over the last decade or so is a vast array of new treatments, so mainly biologics, which are just incredible in their ability to change the immune system, which is the main driver of disease in IBD. But we now have probably a dozen or 2 of these biologics across different immune-mediated conditions, such as rheumatoid arthritis, multiple sclerosis, and IBD, not to mention the use of biologic in cancer therapy as well, which is just fantastic. But our ability to decide what to use when, and I'm just showing an example of an algorithm on the top right that tries to identify what we do with a patient, is a huge challenge for us. And we're learning this and changing this all the time. And this is, again, a huge area of need where we need help. On the bottom right, what you can see is what's called the therapeutic ceiling, which remains, I think, a representation of the limitations of what we're able to do right now. Every single therapy at most probably allows for 30% long-term remission, which means that our patients are doing well over time, which is certainly not acceptable. And in this figure, I won't go into too much detail, but some of the potential opportunities where we can break that ceiling and reach higher levels of remission are identified. And I think that AI is in a very good position to allow for that. So to summarize the problem, before we start talking about potential solutions, IBD is an example for a complex condition, is certainly recognized by the multi-factors that are involved in its pathogenesis, including genes and environmental factors. This is not one disease, in contrast to exposure causes disease. And I know that this is oversimplifying it. For example, with cigarette smoking and lung cancer, if we eliminate the exposure, then we might reduce certain types of lung cancers. In this case, we don't really know what to eliminate. There are many different factors that are involved with that. And for our convenience, we're calling this group of conditions IBD, or specifically Crohn's disease and ulcerative colitis. But these are probably hundreds of different conditions that look similar and behave in very different ways. And just conceptualizing that, I think, is very difficult for most physicians and certainly for most families. There's a long lag time between the exposure and the outcome. So in pediatrics, that lag time is a bit shorter, which is why I think that in the area that I'm in, we have more opportunities than possibly in others. But still, we think that events that happen around and before pregnancy probably have an impact, and early life is probably dramatic for that. So that's an important point. There is no good gold standard for the diagnosis. It's a combination of different things. So defining whether you have the condition and what type condition is still a very challenge. There are multiple classifications that we use, and classifications is important because that's the language that we use to speak to our colleagues and our patients, but we really don't have the tools to define this very well. There's huge variability in disease progress. So if I could know at the beginning that one patient is going to do very well on one therapy, whereas others are going to escalate and require surgery again and again, I might make decisions based on that. And certainly that's not unique just to IBD, but it's very dramatic there. And the therapies I discussed about, and obviously the growing burden of disease as this becomes a much more prevalent thing, really raises huge challenges for us all. So sorry to depress you, but I'll try and offer some optimism because I think there are some opportunities that we are recognizing. And it's a bit sad to kind of note the first one, because this is a disease in evolution that is becoming more common. That's not a good thing, but anything that's evolving and changing offers opportunity for us to understand the process, right? And this is where, and I won't talk about this, I think diet probably has a major factor, and we're trying to understand how the change in other environmental exposures over time might explain this. So it is an opportunity to answer some questions around that. Looking for exposures, I think very much relates to that. This is an area that is receiving more recognition and more research support. And I'll talk in the next slide about the engaging environment that we have within Canada specifically. We do have a large amount of data and biosamples, which again, I'll show as examples that I think can help solve some of this. And heterogeneity is certainly a challenge, but it also is an opportunity because we can then better try and classify, for example, with principal component analyses and other ways, which subgroup is clustered together and might behave in a way that we can better identify and define. And then I already mentioned some of the pediatric specific points. One of them is that there are less comorbidities, so it's a bit of a cleaner population. For example, cigarette smoking is a very major risk factor for Crohn's disease, and children are not exposed to that as much as in adults. So what I'm involved in, and this was kind of mentioned in the introduction, is the Canadian Children's IBD Network, otherwise known as KidsCan. It's unique in the way that all of the pediatric academic centers in Canada are involved in recruiting patients to this network. It was initially funded by a BC foundation called the Child Foundation, and we just got a second round of the grant, which was really there to try and create this network where we're now putting our efforts together to tackle different challenges, such as the ones that I mentioned earlier on. We have data and samples from 1,500 children who were an inception cohort. So they were at the diagnosis recruited, and we were able to collect systematically and prospectively their data and samples, which is a very unique resource. So this is something that Canada is the only country to have across the world. And there are multiple collaborative projects in different areas that have allowed for this. And one example that I'll show over here is a team grant that I got and I co-led with Bruce Valens from University of British Columbia, where we are interested in understanding the role of the microbiome. This is work that's just going to be starting. So I hope that we will be using some of what we're starting to work on now, some of the AI techniques, in order to better get the most out of the data that we're interested in collecting. I will spend most of the rest of the time on a couple of projects that I have two students now that are starting to work on. The first one is to assist with treatment decisions. The second one is to improve diagnosis. But I have a very, well, maybe not so hidden agenda, but a different agenda above that, because as a biologist who's interested in pathogenesis and the role of microbes, et cetera, I'm hoping that this can advance basic science, which is maybe a bit different from some of the other talks that you've heard up to now. So the first project really came from this idea, a hypothesis that we had a few years ago, that your previous diet will impact the gut environment and gut microbes, and that may have an effect on your probability of responding to therapy. I think that kind of makes sense. But what you see in the orange boxes are the different tools that we have in order to measure every one of those stages. So FFQ is food frequency questionnaires, which are really quite poor assessments, but there's some of the best that we have of what people eat. And then we can look at the microbes and their products, which I'll discuss very briefly later on. And the clinical outcome with the cohort that we have, we're able to look at in detail. And this was funded by the Imagine Spore Network. So the clinical intervention or treatment that we're looking at is exclusive enteral nutrition. This is unique to pediatric Crohn's disease. I won't go into much detail because of time, but for 6 to 8 weeks, these children stop eating normal food and they're exposed to some of the formulas that you see over here. And remarkably, it has a very dramatic effect. It's effective in about 80 to 85% in getting them into remission. It's not very sustainable beyond that. We have to put them on other therapy, but it is considered the first line therapy by the guidelines that we follow for the treatment of pediatric Crohn's disease. However, as you can imagine, this is difficult to do. And this is a survey that we published where we showed that adherence or keeping up with the diet is extremely challenging across the world. So we want better ways to identify who's going to respond and give them the right treatment. The other point is the multi-omics in the field of GI and the microbes, I think is something that many people have heard about, but there are many other omics as well that we have access to now. And this is really very great in producing data. And this is just showing how we get that. So for example, 16S ribosomal RNA sequencing of microbes gives us a composition of the microbial community. We can look at metagenomic analysis, which gives us compositional data, but also functional data. So what are the genes that these microbes have and are able to express? And then metabolomics, which gives us the actual endpoints of the metabolic process that happens in that setting. All these data together are able to produce some information. This is a paper that we've completed and we're now working on to publish, showing that relationship between the microbiota, your previous diet, and the different outcomes. But to me, it really highlighted the deficiencies we have in dealing with the complexity of data and what we need to do in order to overcome that. And this is where the first project that I'll describe fits in. So a PhD student in my lab, Ricardo Suarez, who's sitting over here, is now in the beginning of his third year of his PhD. And you have the title of his project over here. So he's hoping to apply machine learning for clinical decision support and treatment of newly diagnosed children with Crohn's disease. And our main interest is using the network that I described, where we have 300 children who are diagnosed with Crohn's disease and receive that exclusive enteral nutrition treatment. But we also have a lot of biological data available that and again, we don't have results to share yet, but we will be applying different approaches, starting with the unsupervised machine learning to look at clustering, supervised machine learning, which I'll talk a bit more in the next slide, and then evaluating the classifier that we hope to identify in order to build a platform that we then can apply for different conditions. And this is just to introduce the concept which I think is probably familiar to many of you. So we do have a training cohort, which consists of most of those 300 patients, where the data will be put into a learner to build a classifier that we will then test the performance of with novel individuals that will come through. Now, what we're hoping to do is, and this is again my hidden agenda as someone who's interested in the biology, also fit in a lot of the pathophysiology that will be measured by the microbiome data and metabolomics. This does introduce challenges that I'll discuss in one of the next few slides, but this is where I hope that we'll be able to not only identify the patients who may benefit from this, but also try and get some hypothesis generating evidence for the pathways that might be involved that we might be able to interfere with in different ways. The second project is looking at endoscopy. So colonoscopy is the one we're looking at specifically, and this is an image that you will classically see. This is a review paper that was just published in Nature Review Gastroenterology. There's a lot of interest in this, and similar to many other fields, there are many more review papers than original research papers on AI and in GI in general, but certainly around endoscopy, there's a lot of interest. So when I do endoscopy, I collect information about my patients, and that helps me understand what diagnosis they have. We have different classifications and scoring, and those are the points on the top that we are hoping that machine learning will help us get in a more clear and well-defined way. But I want to focus on the ones on the bottom, because I think that this is where we can get added value beyond what we're able to do right now as skilled gastroenterologists. So if a trainee is using AI as an assist tool to identify disease and say to him, there's something that you missed over here, that is very helpful. But what I want to do beyond that is try and identify areas or information that we don't even know to look at this point. So for example, again, going back to the microbiome, microbes live in the gut in many cases in what's called biofilms. So these are communities where they kind of grow together and protect themselves from the environment. We can't physically see this, but I'm quite convinced, and there's one paper supporting this, that on endoscopy, there are changes in the image quality and appearance that will reflect these changes in the microbial community. So things that we can't see with our naked eye, I believe that the machine will be able to see. And that's one of the goals that we have in the next project. And this project will be led by Bishoy Aziz, who just starting the second year of his PhD. And he is supported by Jacob Jaremko, who's a radiologist, but with a PhD in AI. So this is again, and I didn't mention Russ Grainer in the previous one, who I think many of you know. I have to be supported by AI experts in order to supervise these students, but together we're hoping to build these bridges. Kumar and Abhilash are very instrumental in building some of these instruments together. And this was just a pilot study that Bishoy already did using about 1,500 images from 200 studies that we have to try and build the models. And a lot of this is neural networks that you heard about before. I'm not a big expert in what this actually involves, but I think that the concepts are important. What we're hoping to do is three things. So first of all, as a proof of concept, show that the machine is able to do what I'm able to do, and that's identify the disease. And we're going to do this with videos, which will provide us with a much larger amount of information than just the images, of course. We hope to then use those data to predict therapy response. And again, it's nice to have the two projects linked, but we're using very different data to do that. And then the third point is what I hinted to before, to try and link the details in the images to possible pathways and pathogenesis that we just don't have the ability to do. And what we're working on now, so we have about 500 videos of children with ulcerative colitis at time of diagnosis. The main challenge that we have, if you look at one of these videos, is that there's a lot of garbage. There are a lot of images there that are useless, where there's either liquid covering the camera or you're looking at the wall, you're not getting clear image. So our first task is to try and identify readable images. And the way I like to explain this is, I happen to be a hockey fan, and I like watching the full 3 hours of the Oilers games, but sometimes I just don't have time. So I watch the condensed game, right? That's about 8 to 10 minutes or so. We want to be able to produce that from a full colonoscopy, which is half an hour to 45 minutes, in a way that will be much more useful for us. That's kind of our first task. And only once we achieve that, we'll be able to start looking at what these images are showing us and what findings might be there. We're armed by people like myself and a few colleagues who are experts and are able to do the annotation of these images and videos. So we have to teach the machine what is okay and what isn't, what is disease and what's not disease, etc. And through the network, there's actually an endoscopy committee, which is very much involved in collaborating on this. So what are the challenges that we have? And, I think, again, I'm at very early stages, so I hope to receive feedback either now or during our break from any one of you. But certainly there are quite a few challenges, and it didn't take me a long time to come up with this list. I probably could have come up with more, and I'm sure that you can all come up with more. But we don't have enough patients with good data. And we're in a situation where I really like that the graph that Dr. Wong showed about, the quality of data and the number of data. So we are in a position where we probably have good quality of data, but our numbers, 300 to 500 sounds like a lot, but I think in the machine learning world, it's really not enough to make good conclusions. And we, in that sense, have more features and subjects, especially if our goals are using multi-omics. And I think, we can overcome some of this, but I recognize that it is a problem. With that is the risk of overfitting, and obviously, deciding how we build all our algorithms around that is important. The disease heterogeneity introduces the risk of signal to noise ratio. So, if we have something that's very clear and strong and is very uniform, we are likely to identify that signal. But here there is going to be a lot of noise. And I think that using a two-tier approach where we first divide the groups and then look within the group and try and understand that signal might be the right approach to overcome that. Something that's true for any observational research, we have the unknown and unmeasurable factors. So I know what I know, but I don't know what I don't know. I'm sure you've all heard the different variations of that. It's so true for the work that we're doing, right? And we look at what's on the spotlight, but there are lots of things that we just don't know to look for. Details and time series cohorts is lacking as well. So ideally, if we could have a time course longitudinal study where we're collecting things over time and looking at change, that would be great. Some of our data and samples do allow for that, but most of it is more cross-sectional. Not all the measurements that we're looking at are standardized and reproducible, and probably one of the best examples is microbiome research. It depends on so many factors: how the sample's collected, how it's kept, frozen, measured, which computing algorithm you use. So we have to overcome that as a community and make sure that we're standardizing all of that. And one of the problems that I've needed to deal with is that this is not intuitive for someone like myself who has, learned some primitive statistics and is used to looking at regressions and more standard ways of looking at things. And just saying that, the computer is telling us that this patient needs to see a cardiologist, I want to know why, but it's in the black box. We can't, this is what the data are showing us. So it's a concept that I think the cultural change within the medical community really needs to be affected by that. So we certainly need help with this. So just to summarize, there's a clear need for innovation in this field, and I'm very delighted to see all the people over here. Big data are big opportunities, but with big challenges. Machine learning offers an attractive option for overcoming some of these challenges. And I think that, combining the different types of data is one way of doing that. Getting the most of the images is the second example that I showed. Examples of integrative science with much room for collaboration, I think is what we're hoping to achieve by this. And I'm really looking forward to your feedback. I just want to thank the people in my lab on the left, the collaborators, especially those kind of shown in the bottom who are within the AI world. And again, a picture of my lab on the left and the clinical colleagues of mine on the right. And thank you so much for your attention. I know that we're into the break time and I hate to do this, but do you, yeah, questions? Yeah. Questions, anyone? Sure. Yeah, so just for people online, I'll repeat the question about swallowable pills with electronics. They're able to measure different physiological features within the gut. And that's a great example for newer technologies that are coming through. So we've been using for many years capsule endoscopy, which takes images in the bowel that you can swallow, but there are much more sophisticated and even robotic technologies that are coming through. There are nanotechnology options that are increasing measurement. And I think that this will just really add to our knowledge in a dramatic way. It's not something that I have personal experience with, but I think it's another example for the challenges that we'll have in dealing with all these data that we're going to get and learning which research questions we have to ask, because technology is moving forward very dramatically and quickly. I'm just not sure that us as researchers and clinicians are prepared to deal with all that. So there's a lot of work for all the young people here to do over the next few decades. Thank you for that. Sure. Yeah, so again, thank you for that point, talking about the gut-brain axis and the strong links between really different systems and organs in the body. And I think so there are more nerves going to the gut than any other organ in the body, including the musculoskeletal system. And it is a bidirectional relationship. Microbes are very much part of that. Hormones are affected by that. And I think it just adds another level of complexity. Patients tell us about this association and, the fact that stress, for example, can have an impact on your system and the course of disease. It's definitely there. And, I'm glad you mentioned that, because it's an important part of what we deal with. And again, adds to the complexity. And now that, our neurophysiology is advancing, imaging is advancing, there are great opportunities to look at that level as well. Thank you. Break time.